Yeah, I'll just go to the slide. So it's in slideshow now for everybody. Is that okay? It's in slideshow mode, right? Okay, great. So we'll get started. So I think the topic, my topic today is defining and aligning the study objective with the methodology components. So it's a very broad area, and I know you are going to have specific lectures which will be reflecting on each of the section in the method. So what I'm going to focus on really is the structure. So even as a reviewer, when we see proposals, quite often we find that certain sections itself are missing, even at the stage of peer review, when we get a full article. So ideally, if you have written your proposal well, you should have very little work to do when you're writing manuscript as far as your method section is concerned, because most of the things which you have written will, as it is, become your manuscript. So I will focus a lot on what section should be there and what need to go into those sections, because that was a topic given to me. And also try to, talk about few examples, and I'm also happy to hear from you on this. So the competency to be gained is how to write protocol for an epidemiological investigation in aligning objectives to the methodology. So I think most important thing is really articulating your objective well. And if you have articulated your objective well, then you will be able to make the connection very well with the methodology. So depending on, like, I come from an epidemiology background, so I'm using some epidemiological examples, but if you are coming from the lab background, you will have certain norms as to how to frame objectives, right? So main thing is that you need to use the appropriate language while writing your objective. And it's not just for the language perspective, it's also because it is connected to your study design and your population and all of that. So when you use the epidemiological language, it in a way, you are also indirectly telling whether you are doing a descriptive study or you're doing an analytical study. So first of all, when you write your objective, you need to make sure what is your primary objective, because your primary objective then will align to your sample size. And I know you have a session coming up on sample size. So quite often people will have multiple questions. You may have multiple questions, but you will not be able to calculate your sample size for everything. And therefore, defining what your primary objective is extremely important to align you to the various sections of your methods. And you need to be very clear whether you are testing a hypothesis, means is it analytical, like you are answering a question on risk factors and so on, or you are measuring a quantity, like prevalence of something, right? So that is measuring a quantity. So now coming to, so our focus is going to be how do we align objective to the various sections of methods in your proposal. And so these are the 10 sections that we use as a standard, in our MPH program and in our various training programs. And therefore, I've used this framework, and I think it will work for most of you. So as you can see here, there are 10 sections, starting with the study design. And if you are doing an intervention study, there will be a section on description of interventions. You have study population, operational definitions, sampling procedure, sample size, data collection, analysis plan, quality assurance, and human subject protection. I'll be spending little time on each of this, and also try to tell you, depending on what your study objective is, what may go into that particular section. So first is study design, and I know there's going to be another lecture on that, but I would like you to just think through that once you have decided what your objective is, you need to think, these are the commonest study design. Are you going to do a cross-sectional survey? Are you going to do a case control study, or are you going to do a cohort study? Of course, you may be doing a randomized control trial if you are in the clinical research side, or if there are even community-based randomized control trials, which you may be planning. But I know as far as we see most proposals which come to ICMR for review, they sort of fit into one of these categories. And you need to think how this objective has led to this particular study design. So I'm just going to pause for a minute. If any participant want to put in the chat box, I'm going to say, let's say your objective here is to estimate the prevalence of diabetes in X district. So what will be your study design? Anybody want to put it in the chat box? Very good. Somebody wrote cross-sectional. No, this should not come from the scientist. I was expecting this to come from other people. But anyway, now you have a question. Let's say you want to understand the risk factors for oral cancer in X district. What will be your study design? You want to understand the risk factors for oral cancer in a population. Okay, every possible design has been written there. So you cannot do cohort study. It's extremely difficult to do a cohort study to answer the question on risk factor for a rare disease. So I think you will be hearing more about that. But when you are dealing with a rare disease, if your objective is to know risk factor for a rare disease, your study design will be a case control study. But if you want to study multiple risk factors and multiple outcomes, but then they should not be very rare, then you could do a cohort study. Definitely not cross-sectional study. So in cross-sectional study, what we call as the chicken and egg story, right? So you don't know which came first, whether the risk factor came first or the outcome came first. And hence, you could do like what we call as analytical cross-sectional survey, but in terms of it's less, it will give you a less accurate information compared to doing a case control study. So aligning your objective to your study design, I think that is the first most important thing, because all the other sections of your methods will be linked to that. Now, quite often what happens is that you have already thought through your questions, but you have many questions in mind. And that's quite common situation. Like, I also want to know burden. I also want to know risk factor. I also want to know where they take treatment. I also want to know cost of care. So if you have multiple questions, you need to remember that one study design may not be able to answer all the questions. Sometimes it also means that you need to do many studies. Or sometimes it means that you need to have many components, and each component need to be sort of a separate section on its own. So for instance, I can give you an example here. If you want to know the burden of oral cancer, how will you know the burden of oral cancer? Anybody want to put it in the chat box? Okay, somebody said descriptive, cross-sectional. Will you do a study in the first place? Is doing survey the best thing for oral cancer? Will you do a survey at all? Very good. Someone wrote, you will do a registry. So if your question here is literature review, of course, if the data is there, right? If the data is not there. So we can have many research questions. The point is, are you picking a research question which is connected to the study design, and whether that study design is feasible and practical in your setting? So for instance, if your objective is to actually know the burden of oral cancer, you should not do a survey. You should set up a registry, which is a totally different methodology compared to doing a survey. So that's the point I'm trying to make. So once you have thought through your what your question is, you need to align to what kind of design you are going to do. Now coming to the next section of your method. So quite often people do interventions. It could be a clinical trial, or it could be a community-based intervention. And when you want to describe in your results section, so if it's a RCT, there are pretty standard guidelines how method section of RCT should be written, and in terms of identifying the treatment group, control group, and so on. But your intervention could be in a community setting or in a primary healthcare facility and so on. And in those situations, you will be looking at defining your intervention, describing it, to whom you are giving the intervention. What is the intervention? What are the various components of your intervention? When will you do it? At what time point? How long is your intervention? For example, one of my students was recently doing a study on how counseling somebody, coming to a primary or secondary healthcare facility on use of salt will actually change their intake of salt, or at least awareness about salt, or they will add less salt or eat less salty food. So if that is what you want to do, then you need to describe that who will be your patient, how long will you do the counseling? Is it only one time? Is it multiple times? Is the patient going to come back once, twice, thrice? How many times you are going to sort of take the follow-ups and so on? So you need to describe all that in detail. How long will be your intervention? When it will be given? How it will be given? So it could be in a trial, it could be a medication, or it could be some other kind of intervention, or it could be behavioral intervention. As I said, it could be counseling. It could be, so what are the other common intervention you think you may be? Anybody want to put in the chat box? What are the common intervention you think you may be doing in the kind of proposals you write? Any thoughts? What kind of intervention studies you may be doing? I understand many of you are investigators, and that's why you are part of this workshop. Anybody has any thoughts on any example of intervention? Diagnostic. Diagnostic means you are doing diagnosis of a disease, lifestyle modification, treatment. Okay, we already talked about this lifestyle example. Drugs. Yeah, drugs is where you will do. Okay, very interesting. Cessation. For instance, you may be doing tobacco cessation or smoking cessation as an intervention. So what does it involve? It means then how will you fill this section? Whom will you give the education? Are you going to do it to patient or to the community? Is it at the level of health facility, or is it the level of community? Is it the intervention given by a trained healthcare worker, or what we call as a lay healthcare worker? Is it an intervention given by a doctor? Is there a special team? Somebody said nutritional intervention. Are you going to give a different kind of diet to them? Then how long will you give? And where they will get that? Is it at home? Is it at a particular place? So all those details need to come into this section. Now comes the most important section, inclusion and exclusion. So whom do we include in a study? So inclusion criteria has to be very clear, because quite often what happens is that you have, let's say if you're doing a population-based study, when you go to a household, you may have people of different age group, different genders, and different characteristics. So if your study is your study in the reproductive age group, is your study for only adults, only for children, and are you excluding within that population someone? Like for instance, some study may say, okay, it's for women, but which are not pregnant, let's say that. Or if it is, let's say it is for people below the age of 60 years. So inclusion criteria really need to define what age, gender characteristics, and if there are any specific type of group you want to study. So there are certain studies you may do, let's say in certain occupational group. You may say this study will be among people who work in certain kind of industry and so on. So that's about the population. Then coming to the operational definitions, which is very important. You need to really spell out your case definition very clearly. So for example, if your study is on diabetes, you need to say, what do you mean by diabetes? What is the definition you're using? Is it based on random sugar? Is it based on fasting sugar? Is it based on capillary? Is it based on venous sample? So you need to define that, whatever is your outcome variable. You also need to define your endpoint if it is a trial. For example, if you're doing a study on a clinical trial on, let's say, diabetes, then what will you measure in the end? Are you looking at HbA1c? Are you looking at fasting sugar level? Are you looking at behavior change? So that becomes your endpoint. And how, if you are doing a comparative trial like a RCT or even a non-randomized trial, who will be your control group? And when you're doing operational definition, it could be either the standard definition which are there in the literature you could use, or if not, you could also draw from your own experiences. Sometimes you're studying something for which standard definitions are not available, and hence you can draw from the literature, but also from your own experiences. Now, next, this will be in detail explained in the next talk. I'm only giving you the structure. So the next section needs to be about the sampling procedure. When you say sampling procedure, what does it mean? Are you going to conveniently pick a particular group of people, or are you randomly selecting the sample for which you need to have a sampling frame? Or are you doing systematic sample? Or are you doing a cluster sampling? Quite often in public health, we use cluster sample, means we take, let's say, a ward or a village as a cluster, and then within that we do various levels of sampling. But in clinical setting, it may be very different. You may take all the patients, let's say, walking in from 1st January to 31st January with XXX diagnosis will be included, which is fine too. And if you have any references as to why you think this is the best design, you can give those references. Now, sample size, again, this how to do sample size, I think there's a separate talk on that. So I'm only going to tell you what should be written in the proposal. You need to give all the parameters which are used for estimation of sample size. And most importantly, you need to know, you should do very good literature review for that. You should see, if suppose this similar study has been done elsewhere, what kind of sample size they have used and what was their outcome. So let's say if your study is on prevalence of diabetes, just giving you a very simple example, you have to actually tell your statistician, or if you're doing it yourself, now there are OpenEpi and other software, you can do it yourself as well. But they will ask you, is the expected prevalence 8%? Is it 15%? And then you say, no, I don't know. But then you need to find the closest reference. Has it been done in a similar district, in a similar state? So you have to take from that and then say, okay, I think it will be somewhere 8 to 10%. And then based on that, the sample size is calculated. Now, once you have done this, these are the really important section, and then comes what data you are collecting. And this section should have what data you are collecting, from whom you are collecting the data, how you are collecting. So you may be, let's say your data collection could include a questionnaire. It could include measurements, like you may do anthropometry, you may do lab investigations. And then how will you collect? Where? Is it a community-based data collection? Is it facility-based data collection? Who will collect? Are you going to train some particular staff, or so on? And then you have to describe what instrument, and these instruments go as your annexures. So you're using any questionnaire, abstraction forms, any observation guides, and so on, and then give them as annexures. And analysis plan, that is the next section of the method section. And quite often people say, no, we will do analysis once the study is done. No, it cannot be done like that. You have to think through your analysis. And in fact, that is the most important thing. Sometimes it may even change your whole study. When you get into analysis, you realize, oh no, I can't do this. Then maybe I should do this study differently. Which software you are using, what variables you have, what are the measures that you will be actually estimating? Are you going to estimate a prevalence? Are you estimating an incidence, odds ratio, relative risk, and so on? And then you need to also think how you will do the quality assurance in each of these sections. So I know there are separate lectures on these topics, but I just wanted to give the structure. So there will have to be one section on human participant protection. And then you need to also attach all these data collection instrument and all as annexures. So I want to ask the organizer, if my time is over, I'll stop here. Otherwise, I can run through an example.
You have 10 more minutes.
I have time. Okay, great. So I'm going to run through one example, and I would like, if people have any specific question, and those can be posted. So sample size estimation, someone said that's not part of this lecture. There will be a separate section on that. But apart from that, I request investigator to post questions. Okay. Specifically around the structure. So let's be clear, we are talking about the structure of method section here. And the main idea is on how you align your objectives. So that's what I'm going to walk you through one example now. So this is again, I've taken a real study done by one of our MPH scholars to estimate the proportion of missed visit among hypertension patients who were on treatment in a primary healthcare facility in XX district in the year 2022. So that's the primary objective. And the secondary objective is to describe the reasons for missed visits among individuals with hypertension who are taking treatment. So first question I want to ask all those who are listening is, what do you think should be the study design? Can you put it in the chat box? what do you think should be the study design? Anybody? Very good. Somebody said cohort. Somebody said cross-sectional, descriptive. Okay, very good. Okay, so cohort or cross-sectional, right? So here the idea is to estimate the what percentage of people who are taking treatment have actually come to the facility. Okay, so now if you already have the line list available, so I'll come to that. So there you can do both. So if you don't have any data available, you will actually need to do it as a cohort study. But if you do have the data available, then you could also get this information through a cross-sectional survey. So, sorry, slide is not moving. Yeah. So this was a cross-sectional study. And so this is here, the setting is that this is a primary healthcare center where the people are taking treatment over a period of time. And then you want to know what proportion of them came back or not. So at one time, you go and you review the records and you look at people, all those people who have high blood pressure, and then you calculate how many of them actually have come back or did not come back. So the operational definition here would be you have to see who is a hypertensive. So whatever is the methodology used in your protocol. So mostly we know that it is more than or equal to 140 or more than or equal to 90, or people already who are taking medications. And how to do this study? So this student wanted to do it in multiple facilities, but then we have the list of facility available with us. So that makes it easier for us, and because we can make a list of facility, and then within that we can again also take a sample of patients. And this is a setting where the line list is already available. Like we already know these are the 20 patient facility A, or these are the 30 patient in facility B. So the data was captured from the treatment cards. So the card is maintained in the health facility. And then you could go and read from that and make out whether this patient has come back or not. And then for once you got the list of people who didn't come back, this student also called the patients over the phone and to answer the second objective, which is to understand why they did not come back. So they made a phone call and asked them, okay, where did you go? Did you come back? Did you go to another facility for treatment? Are you continuing treatment, or have you stopped the treatment? So then analysis plan need to tell what the proportion is with 95% confidence interval. I think that's very important. You always need to give your estimates when you estimate is an outcome with 95% confidence interval. And then they talked about what were the described what are the various factors. The protocol was approved by ethics committee. So I think I'm going to stop with that. So just to summarize, always start with the concept paper. So the sections that I mentioned you should write down these sections and start filling them up. So once your objective is final, just take a one-pager and write down all these subheadings and start putting at least one line under each one of them. And as you go along and you brainstorm, you do the peer review, you keep expanding each of these sections so you get more clarity about each of your objectives. So I'm going to stop with this so we have more time for discussion and questions. Thank you. I hope I covered the topic because it was very broad. So I try to kind of summarize.
You have covered it very nicely. Like we were needing the 10 pointers what you have given. Like one is writing the how what should be the points and how it will be aligned with the objectives. So now we are having a few queries. So we have a time of 5 to 10 minutes, so we can take these queries.
So I would really like the participants to focus on these 10 points when they're writing the method section. And any questions around that and aligning objectives to this?
We have a few questions in the chat box. And first one is, how can we calculate the sample size for the novel study, or where you don't have the prevalence or literature data?
Yeah.
So how can we calculate the sample?
Yeah, I mean, yeah, that's a little bit beyond the scope of this lecture, but I can answer that. So when it is a new condition and you need to do a pilot. So if you don't know at all, then you may need to do a pilot. So you may need to do a smaller study before you actually jump on to a bigger study. But quite often, it's very rare that such a thing happens. You will have some estimate. It may not be there from your own sort of, let's say, district or state. It may be from another state or even from other countries. So and if you don't have any data at all, then do a small pilot. That's the best way. I think there's a question on time gap between the. Okay, how would you define missed visit? Okay, in this particular study, the definition of missed visit was the people, like there was a quarter which was defined. If they didn't come during that particular quarter, let's say from 1st January to 31st March. But you can have your operational definition, whatever is suitable for your setting. Pilot need sample size calculation. I think you need to be specific. I'm not able to understand, because very rare that you have no idea at all about how common a condition is. So it's a little difficult unless you have any specific example that I'll be able to give how to move forward on that.
We have a question.
Any pilot for clinical trial. Pilot for clinical trial. See, usually when you put something into a clinical trial, it would have gone through your phase 1, phase 2, right? So why would you do pilot? You go through the phases of trial. So you already know that the intervention has been tested under the control conditions.
You showed an example of 384 samples, like, CI 95 and 50%, I mean, I was just wondering, did you keep the timeline? Because projects are usually, time bound. So 384 for how long? What should be the timeline to achieve these 384?
Timeline for?
I mean, for sample calculation, once you are calculating the sample size, so you showed an example wherein, you need to have a CI 95, and then you came out with the numbers 384. So for how long we have to collect the 384?
How long you are saying it will take to collect 384? See, the thing is here, it's a setting where the data is being extracted from records. So in this particular study, it was extracted from records. So it's really how much time, how much staff you have to do the data extraction.
Yeah. For a year,
but if you're doing a survey, I mean, in this study, this was not meant for a survey. This was to abstract data from records. But if you're doing a survey, then it really depends on how long, I mean, how many staff you have and how many people are collecting data, right? That determines how much time it takes to complete any study. I think sample size, I would request you to hold your all the questions for the next session. So if we can focus more on the structure here, because this particular session is not for sample size. Yeah, I know all the questions are on sample size. I request organizers to maybe copy these questions and share with, I think Dr. Pandey is doing the session on sample size.
Someone has asked about quality assurance, like we have not discussed much.
Yes, good. That's a good question. How do you do quality assurance? So quality assurance has to be done at all levels. It can be starting from, when you're writing protocol, you can do quality assurance by doing a peer review. Like if you share with people, who have worked in that area, they give you comments. That's the first step. Second is for your data collection. So when you're doing data collection, the quality assurance has to be done depending on where the data is collected. Or if it's a lab, then you need to have the lab quality assurance method. But typically in epidemiological study, what you can do for quality assurance is, one is you train your staff well. You try to minimize variation between the investigators. They all should understand or ask the questions in the same way. That's why we say, like, your question should be spelled out. It should not be like, one words. You spell out complete question and wherever possible, translate it into the local language, because that ensures that the questions are asked same way by all the investigator. The other thing in large surveys, what we do is you can also do what we call as in a sample, you can do a resurvey. So for example, in large surveys, you may say the survey has, let's say 5,000 participants, but a small percentage of that, let's say 500 or 1,000 or whatever, you fix what is feasible, you send another person to the same respondent and ask at least 3 or 4 key questions and see whether the answer given before and answer given in this quality assurance survey is actually matching. So that is like for the bigger survey that is done. The next level you need to do quality assurances at the level of data entry. So if you are doing, nowadays a lot of surveys are done using Palm Top or Android phones and ODK and so on. So that way, in one good thing is that itself does quality assurance, because you can set fields in a certain way. For example, if your age group is, let's say, below 60 years, and you can set field in such a way that above 60 years cannot be entered at all. So that way you can do quality assurance at the data entry level. But if you are doing, let's say, paper-based entry, then you can do double data entry for the quality assurance. And the next level, it's at the level of just data cleaning and analysis. So at each level, you can do quality assurance. And someone has rightly mentioned that if you are doing a lab study, you need to write a separate section on how you will do the quality assurance for that particular test. So it could be different ways of doing quality assurance in different settings. But like I can tell you from my experience, for instance, in some of the studies that we have done, for example, CMC Vellore has a quality assurance program. Like, so for if you want to do a particular test in a lab, let's say even sugar, you can tie up with these quality assurance programs, and then they will be taking they'll be sending a sample to that lab and then telling you whether the value is within acceptable limits or not. So if there are any other questions.
We have last question here.
Dr. Prabdeep, I am Geeta Menon here, and it was a wonderful presentation that you made. I have asked you your take on, we are talking about the quality assurance of a protocol here, right? We're not talking about quality assurance at different stages of the study. But if we are looking at the quality assurance of a protocol, what is your take on publishing the protocol? Once I know that it goes to a funding agency, it is reviewed by experts. But do you think publishing the protocol before you actually do a study increases the quality assurance of the protocol?
Yeah, it gets peer reviewed by, external peer reviewers. So definitely, but that's feasible only for, I think, bigger studies, and also for maybe more mature investigators, who know how to write protocol for a journal. But if you are a relatively early career investigator, it may be difficult to publish.